हेलो लर्नर्स, वेलकम बैक टू आवर चैनल बी क्यूरियस। आज हम पढ़ने जा रहे हैं प्यूरिफिकेशन ऑफ डीएनए फ्रॉम लिविंग सेल्स। इसमें हम कवर करेंगे प्रिपरेशन ऑफ टोटल सेल डीएनए, प्रिपरेशन ऑफ प्लाज्मिड डीएनए, प्रिपरेशन ऑफ बैक्टीरियोफाज डीएनए। द जेनेटिक इंजीनियर will different times need to prepare a list three distinct kind of DNA. First, total cell DNA will often be required as a source of material from which to obtain genes to be cloned. Second, total cell DNA may be DNA from a culture of bacteria, from plant, from animals, or from any other type of organism that is being studied. It consists of the genomic DNA of the organism along with any additional DNA molecules such as plasmids that are present. The second type of DNA that will be required is pure plasmid DNA. Preparation of plasmid DNA from a culture of bacteria follows the same basic step as purification of total cell DNA. Finally, phage DNA will be needed if a phage cloning vector is to be used. Preparation of DNA cell preparation of total cell DNA. The fundamentals of DNA preparation are most easily understood by first considering the simplest type of DNA purification procedure. The procedure for total DNA preparation from a culture of bacterial cell can be divided into four stages. A culture of bacteria is grown and then harvested. The cells are broken up open to release their contents. This cell extract is treated to remove all components except the DNA. The resulting DNA solution is concentrated. Growing and harvesting a bacterial culture. Most bacteria can be grown without too much difficulty in a liquid medium broth culture. The culture medium must provide a balanced mixture of the essential nutrients at concentrations that will allow the bacteria to grow and divide efficiently. M9 is an example of a defined medium in which all the components are non. This medium contains a mixture of inorganic nutrients to provide essential elements such as nitrogen, magnesium, and calcium, as well as glucose to supply carbon and energy. In practice, additional growth factors such as trace elements and vitamins must be added to M9 before it will support bacterial growth. Luria botany (Lb) is a complex or undefined media because two of the ingredients tryptophan and yeast extract are complicated mixture of non-chemical compounds. Tryptophan in fact supply amino acids and small peptides while yeast extract provide the nitrogen requirements along with sugars and inorganic and organic nutrients. Complex media such as Lb needs no further supplementation and support the growth of wide range of bacterial species. Preparation of a cell extract. The bacterial cell is enclosed in a cytoplasmic membrane and supported by a rigid cell wall with some species, including E. coli. The cell wall may itself to be enveloped by a second outer membrane. All of these barriers have to be disrupted to release the cell components. Techniques for breaking open bacterial cells can be divided into physical methods in which the cells are disrupted by mechanical forces and chemical methods where cell lysis is brought about by exposure to chemical agents that affect the integrity of the cell barriers. Chemical methods are most commonly used with bacterial cells when the object is DNA preparation. Chemical lysis generally involved one agent attacking the cell wall and another disrupting the cell membrane. The chemicals that are used depend on the species of bacterium involved, but with E. coli and related organisms, weakening of the cell wall is usually brought about by lysozyme, EDTA, ethyldiamine tetraacetate. Lysosome is an enzyme that is present in egg white and in secretions such as tears and saliva, and which digests the polymeric compounds that give the cell wall its rigidity. EDTA removes magnesium ion that are essential for preserving the out, overall structure of the cell envelope, and also inhibits cellular enzyme that could degrade DNA. In this structure, it's showing preparation of cell extraction. A section is a cell lysis. Here, cell are break down into pieces, and B is centrifugation of the cell extract to remove insoluble debris. B structure showing centrifugal to remove cell debris. So EDTA is sufficient to cause bacterial cell to burst, but usually detergents such as sodium dodecyl sulfate SDS is also added. Purification of DNA from a cell extract. In addition to DNA, a bacterial cell extract contains significant quantities of protein and RNA. A variety of methods can be used to purify the DNA from this mixture. One approach is to treat the mixture with the reagent which degrade the contaminants, leaving a pure solution of DNA. Other method use ion exchange chromatography to separate the mixture into its various components so the DNA is removed from the protein and RNA is in the extract. Removing contaminants by organic extraction and enzyme digestion. The standard way to deproteinize a cell extract is to add phenol or 1:1 mixture of phenol and chloroform. The aqueous solution of nucleic acid can then be removed with a pipette. With some cell extract, the protein content is so great that a single phenol extraction is not sufficient to completely purify the nucleic acid. This problem could be solved by carrying out several phenol extract one after the other, but this is undesirable as each mixing and centrifugation steps result in a certain amount of breakage of the DNA molecules. The picture showing two steps. First is degradation of contaminants, and second is separation of DNA from contaminants. DNA contaminants give rise to pure DNA, and after DNA from contaminants, it's for give rise to pure DNA and contaminants. Second picture shows that removal of protein contaminants by phenol extract. Cell extract mixture with phenol then give rise to separate layer of by centrifugation. First layer is aqueous layer DNA plus RNA. Second layer is interface coagulated protein, and third layer is phenol. Using ion exchange chromatography to purify DNA from a cell extract. Biochemists have devised various methods for using difference in electrical charge to separate mixture of chemical into their individual components. One of these methods is ion exchange chromatography, which separates molecules according to how tightly they bind to electrically charged particle present in chromatographic matrix or resin. DNA and RNA are most negatively charged, as are some protein, and so bind to a positive charge resin. The electrical attachment is disrupted by salt removal of the more tightly bound molecules requiring higher concentration of salt. Concentration of DNA samples. Organic extraction obtained result in very thick solution of DNA that does not need to be concentrated any further. Other purification methods give more dilute solution, and it is therefore important to consider methods for increasing the DNA concentration. The most frequently used method of concentration is ethanol precipitation in the presence of salt and at the temperature of minus 20 degrees Celsius or less. Absolute ethanol efficiently precipitate polymeric nucleic acid with a thick solution of DNA. To the ethanol can be layered on top of the sample, causing molecules to precipitate at the interface. A spectacular trick is to push a glass rod through the ethanol into the DNA solution. When the rod is removed, DNA molecules adhere and can be pulled out of the solution in the form of a long fiber. So this structure showing attachment of DNA to ion exchange particles, and second one is DNA purification by ion exchange chromatography. In first picture, it's showing ion exchange particles and DNA molecules give rise to salt, and this is showing ion exchange particles. And in DNA purification by ion exchange chromatography, it's going through salt solution ion exchange resin and give rise to chromatography, and then more salt added and give rise to DNA extract and protein plus RNA extract, or protein plus RNA extract discard. Alternatively, if ethanol is mixed with a dilute DNA solution, the precipitate can be collected by centrifugation and the dissolved in an appropriate volume of water. Ethanol precipitation has the added advantage of leaving short chain and monomeric nucleic acid components in solution. Ribonucleotides produced by ribonuclease treatment are therefore lost at this stage. Measurement of DNA concentration. It is crucial to know exactly how much DNA is present in a solution when carrying out a gene cloning experiment. Fortunately, DNA concentrations can be accurately measured by ultraviolet UV absorbance spectrophotometry. The amount of UV radiation absorbed by a solution of DNA is directly proportional to the amount of DNA in the sample. Usually, absorbance is measured at 260 nanometer. Other method for preparation of total cell DNA. Bacteria are not the only organism from which DNA may be required. Total cell DNA from the example plant or animal will be needed if the aim of the genetic engineering project is to clone genes from these organisms. Although the basic steps in DNA purification are the same, whatever the organism, some modifications may have to be introduced. to take account of the special features of the cell being used. Obviously, growth of cells is liquid medium is appropriate only for bacteria, other microorganisms, and plant and animal cell culture. Preparation of plasmid DNA. Purification of plasmids from a culture of bacteria involves the same general strategy as preparation of total cell DNA. A culture of cells containing plasmids is grown in liquid medium, harvested, and cell extract prepared. The protein and DNA are removed, and the DNA probably concentrated by ethanol precipitation. In a plasmid preparation, it is always necessary to separate the plasmid DNA from the large amount of bacteria chromosomal DNA that is also present in the cell. The presence of the smallest amount of contaminating bacterial DNA in a gene cloning experiment can easily lead to undesirable results. So this structure showing attachment of DNA to silica particle. So the silica particle and DNA molecule are attached, and going through a salt solution, it is separated. DNA purification by column chromatography. So after the chromatography, DNA denatured by chemicals are discarded. The method are based on the several physical differences between plasmid DNA and bacterial DNA, the most obvious of which is size. The largest plasmid are only 8% of the size of the E. coli chromosome, and most are such smaller than it. This techniques that can separate small DNA molecules from large ones should therefore effectively purify plasmid DNA. Separation on the basis of size. Preparation of cleared lysate. The bacterial cell going through the EDTA lysosome and 25% sucrose and give rise to spheroplast, intact cell membrane, or disrupted cell wall. And this spheroplast going through triton X100, which give rise to cell extract, and this cell extract going through with a centrifuge and give rise to cleared lysate plasmids, large DNA fragments, cell debris. Separation on the basis of conformation. Before considering the way in which conformational difference between plasmid and bacterial DNA can be used to separate the two type of DNA, it is not just strictly correct to say that plasmids have circular conformation, because double stranded DNA circles can take up one or of two quite distinct configuration. Most plasmids exist in the cell as supercoiled molecules. Supercoiling occurs because the double helix of the plasmid DNA is partially unwounded during the plasmid replication process by an enzyme called uh, topoisomerases. The supercoiled conformation can be maintained only if both polynucleotide strands are intact. Hence, the more technical name of covalently closed circular DNA. If one of the polynucleotide strand is broken, the double helix converts to its normal relaxed state, and the plasmid takes on the alternative conformation called open circular (OC). Supercoiling is important in plasmid preparation because supercoiled molecules can be fairly easily separated by non-supercoiled DNA. Two different methods are commonly used. Both can purify plasmid DNA from crude cell extract, although in practical although in practice best results are obtained if a cleared lysate is first prepared. Alkaline denaturation. The basis of this technique is that there is a narrow pH range at which non-supercoiled DNA is denatured, whereas supercoiled plasmid are not. If sodium hydroxide is added to a cell extract or cleared lysate so that the pH is adjusted to 12 to 12.5, then the hydrogen bonding in non-supercoiled DNA molecule is broken, causing the double helix to unwind and the two polynucleotide chain to separate. Plasmid amplification. Preparation of plasmid DNA can be hindered by the fact that plasmids make up only a small proportion of the total DNA in the bacterial cell. The yeast of DNA from a bacteria culture may therefore be disappointingly low. Plasmid amplification offers a means of increasing this yield. The aim of amplification is to increase the copy number of plasmids. Some multi-copy plasmids have the useful property of being able to replicate in the absence of protein synthesis. Preparation of bacteriophage DNA. The key difference between phage DNA purification and preparation of either total cell DNA or plasmid DNA is that for phases, the starting material is not normally a cell extract. This is because the bacteriophage particles can be obtained in large numbers from the extracellular medium of an infected bacterial culture. So this structure is showing preparation of phage suspension from an infected culture of bacteria. And so infected culture of bacteria uh, go through centrifugation and give rise to phase suspend and pellet of bacteria. Growth of cultures to obtain a high lambda titer. Growing a large volume culture is no problem, but obtaining the maximum phase later titer require a certain amount of skill. Preparation of non-lysogenic lambda phase. Although most lambda strains are lysogenic, many cloning factors derived from lambda are modified by deletions of CI and other genes so that lysogenic never occurs. These phages cannot integrate into the bacterial genome and can infect cell only by a lytic cycle. So induction of lambda CIATS lysosome by transferring from 30 degrees Celsius to 42 degrees Celsius. So lambda prophase going through 42 degrees Celsius induction give rise to excites lambda genome, and this is going through cell lysis, and cell is ruptured and give new lambda particles. Collection of phages from infected culture. The remains of lysed bacterial cells, although along with an intact cell that are inadvertently left over, can be removed from an infected culture by centrifugation, leaving the phage particles in a suspension. The problem now is to reduce the size of the suspension to 5 ml or less, manageable size for DNA extract. Purification of DNA from lambda phase particle. Deproteinization of the redissolved PEG precipitate is sometimes sufficient to extract pure phage DNA, but usually lambda phages are subjected to an intermediate purification steps. Purification of M13 DNA causes few problems. Most of the difference between the M13 and lambda infection cycle are two advantages of the molecular biologist wishing to prepare M13 DNA. First, the double-stranded replicative form of M13, which behave like a high copy number plasmid, is very easily verified by the standard procedure for plasmid preparation. A cell extract is prepared from cells infected with M13 and the replicative form separated from bacterial DNA by, for example, EDT, ethidium bromide and cesium chloride density gradient centrifugation. Thank you for listening.